Hi everyone. It's a great honor to give a talk to you guys, and it is unfortunate that I cannot go there. But it's and given the time difference, I decided to give a talk online, pre-record the talk. Okay, so today's topic is developing fMRI-based biomarkers for pain, and I heard that some of you know the fMRI, but not all of you are familiar with the fMRI. So I'm gonna briefly introduce the fMRI and also my lab's approach to study pain. Let me start with introducing the center that I'm a part of. That is the Center for Neuroscience Imaging Research. It's kind of a brand new imaging center in South Korea. So it's been like 5 or 6 years we built this building, and we got a lot of many MRIs. And this is our director, Sungji Kim, Professor Sungji Kim. He was one of the early people who developed the fMRI, and yeah, so they kind of, this was while he was doing postdoc back in the day, and we had a reunion last year, no, 2 years ago, uh, right before the COVID-19 came out. So we had a really nice conference here. And anyway, so we are trying to build a global and international center for doing MRI studies. So we have 4 MRI machines here, like ranging from 3T MRI to 15.2 Tesla MRI. And these two are, we are using these for animal MRI, like mice and rats. We are actually doing a lot of mouse MRI that is really great to do, actually, because we can use many transgenic lines of for kind of brain, like optogenetics studies for mouse, in mouse. And we also have non-human primates in this building, in the building that I'm in right now. And so we can do some brain imaging for using macaque monkeys. So actually I'm kind of writing a paper on the using, I mean, pain fMRI using macaque monkeys. And also we are using 3T and 7T to study pain in the brain, human brain. And this is my lab equipment, and we have some behavior experimental rooms, like for setting for social and behavior experiments. I really want to invite all of you and show my lab, but yeah, but I'm trying to kind of give the feeling of the lab to you using these pictures. And this is, I mean, there is a physiology machine, and also, I mean, the physiology machine, we can get the skin conductance or heart rate, like these physiological responses. And we also have the pain machines. So this is a heat, this is pressure pain machine, and also we have electric shock machine. And also recently we built new taste delivery system, Octaflow gustometer system, where that we can use for delivering the capsaicin, for example. I'm going to introduce the study that we use, in which we used this capsaicin for pain study. And we also recently bought the rapid cough inflator and olfactometer, and so on. Okay, so I'm going to give you really brief, just one-page introduction to fMRI. So fMRI is an indirect measure of neural activity. So if there is a firing in neurons, it exchanges the energy with these other neurons or astrocytes, and there is going to be a neuronal responses, and that uses the ATP and energy. And so the neurons, these cells, need more oxygen, so that increases the blood flow in the capillary. And also the deoxyhemoglobin content is going to be decreased a little bit. So what we are observing through the fMRI, especially the bold signal, blood oxygen level dependence response, is we are seeing the deoxyhemoglobin contents in the blood. And we are kind of looking at the neurovascular response to the neuronal activation. And what we are getting from the fMRI setup, we are getting this voxel activity over time. Even though we are interested, I mean, if we are using fMRI, we are interested in this functional aspect of brain. So we want to get this time series data of the brain activity. So usually this is some signal that we are getting, but given that we are dealing with the 3D structure, the brain, actually we are getting the 3D plus time. So 4D data are what we are dealing with. And why fMRI? fMRI is located in a really sweet spot of this temporal resolution and spatial resolution. So better spatial resolution compared to other imaging methods that has the similar level of invasiveness. So fMRI is not invasive at all. People can just lie down and then do fMRI scan multiple times. So MRI is really has that strength as a non-invasive method. And also, we can do imaging of deep brain structure using MRI. For example, subcortical areas like basal ganglia, thalamus, and brainstem that are all really important for pain processing, right? So fMRI is really good for pain study because the deep brain structures are really important. And here the temporal resolution goes 1 to 10, but it becomes more like short, getting better and better. So in our lab, we are using the TR around 500 milliseconds. So now we are using 400 millisecond, 460 millisecond TR. So every 500 milliseconds, we are getting the brain images. Okay, so it's a great temporal resolution, kind of. I mean, not like super high resolution, but reasonably good resolution. With that, we can actually capture, for example, the respiratory confounding vectors, right? So we can remove these respiratory confounds, and we can actually look at the neuronal activity, neural related signal in the brain with this short TR. And MRI, I think MRI really revolutionized how we study brain and also mind and body. We can see that we can study task-related activity or functional connectivity, like these functional changes due to some mental functions. And also it's good for us to study structural changes as well. So with one MRI machine, we can study these two aspects like structural and functional. Because of that, we can use this MRI as a bridge between the brain and mind, and also the brain and body. So we can actually study the three-way interactions among mind and brain and body using MRI. So MRI is a great tool and gave us the breakthrough to study the brain and human cognitive and affective functions. And recently, fMRI is combined with AI a lot. I wrote AI because AI is a hot word, but we can use machine learning or statistical learning instead of AI. So when fMRI is combined with AI, we can decode what people think or what people perceive. So this picture describes the brain decoding scheme really in one slide. And in the training set, for example, we show these images to people, the participants, and then we collect the data from this visual cortex area. And then only based on this voxel pattern activity, the multivoxel patterns of activity brain signal, we can decode whether this person is seeing shoe or cat. And in the training set, we aim to find the pattern classifier, and then in the testing set, we aim to decode whether this person is seeing a shoe or cat with a new image, unseen image. And fortunately, we saw early 2010 researchers have shown that we can decode what we see or what we dream. So there has been really nice studies on this, and also there have been many studies that showed that we can decode pain if we combine fMRI with AI. So these are two news articles on Nature, and these are really good articles, so you can check them out. Around 5 years ago, I did a survey because the machine learning, the popularity of machine learning in neuroimaging has been really rapidly increased, and I wanted to see how many papers were there. And almost a total of 400, almost 500 studies were published between these 1983 to 2016, and the number has increased rapidly recently. I mean, that's understandable because the use of machine learning has become popular in every field, right? And these are the use of machine learning models in studies. And you can see, as you can see, people have used these machine learning models for to study neurologic disorders like Alzheimer's. The majority of them were on Alzheimer's or Parkinson's, and also psychiatric disorders. Pain is only really small proportion. And majority of papers actually aim to use them for diagnosis, but maybe more useful but challenging topics will be these 25%, like predicting treatment responses or prognosis or subtyping or symptom classification or risk groups. If we know the risk group early on, maybe we can do the intervention early on, right? And among these, we in the paper we particularly focus kind of were interested in these component process markers because this is what we are kind of usually do, and I'm going to explain what that is. In the paper, I contrasted this component process approach to this direct prediction approach, and in this direct prediction approach, I describe this as a grayscale because what we are doing, what researchers are doing in this approach is using this high dimensional feature of MRI, or the high dimensional feature of brain. They really try to reduce the dimensionality into this one dimension of clinical outcome. But in this component process approach, we can actually have the intermediate layer that are the component processes more closely related to the brain mechanisms like emotion, cognition, pain, some more like fundamental kind of component processes. And then using these markers, we can describe these clinical outcomes. So this is like indirect, like hierarchical modeling scheme, and we really aim to develop these component process markers first, and then try to describe these clinical outcomes that are quite complicated. So as an analogy, I use the grayscale to describe this direct prediction approach, and I use the RGB color scale for to describe this component process approach, so that we can have multiple colors to describe this complicated, complex picture of clinical outcomes or functional outcomes. And the other point that I made in this paper was the traditional mapping approach might not be enough to achieve this goal, to develop any markers. That's because the traditional mapping approach is try to answer this question: is there an effect at each voxel or region? So it's kind of aimed for the local mapping, but actually the relationship between brain and function is the many-to-many mapping. So the whole brain actually achieve a function altogether, right? Many functions. So we need the model that incorporate the distributed representations. But in this traditional mapping approach, there's no model for distributed representations. Therefore, we cannot really achieve large effect sizes, and it involves large numbers of comparisons, so massive multiple comparisons, and because of that, the statistical power becomes really low. So instead of the traditional mapping approach, we suggested doing this predictive modeling approach that actually is really simple, just reversing the equation. So previously, the task was X and each voxel of the brain was Y. And now we are using this brain as an X and outcome as a Y. So this is exactly the decoding approach. And the question becomes like this: like, can we model joint contributions of multiple brain regions to outcomes that we are interested in? And with this approach, we can model the distributed representations. Therefore, this is we call brain-wide pattern-based mapping. And given that we can kind of capitalize on the distributed information across the brain, we can achieve large effect sizes, and there is no need for multiple comparisons, and we can achieve high power, statistical power. And also, people have shown that using this pattern-based coding, population coding, we can actually achieve the fine-grained mapping. Another point that I made in this paper was we need a multi-study efforts, not just one study. So in the future we envisioned in this paper was many different labs come up with these candidate models, different models that target one functions. For example, pain. Multiple labs can develop multiple candidate models, and they have to compete each other to show population until we can get the population level generalization. So in this multi-step scheme, biomarker development process framework, there are multiple stages like development and prospective validation and robust replication and population level replications. This is the increasing levels of evidence, the steps. And most promising models should be carried forward. But when we located the models that we surveyed in these multi-stages, we found that a majority of models just stopped after they published one paper. So this is like one publication and done. So it should not be like that. And so this is what we kind of argued in depth in this Nature Neuroscience paper, that the vectors for better fMRI-based biomarkers. So we need to focus on component process, developing markers for component processes, and we need to incorporate distributed representations, and also we need to put our efforts for multi-study or multi-lab testing. From now, I want to introduce how we implemented these vectors for better biomarkers in studying pain. I want to start with the quote from Francis Collins, who is an NIH director.
We need biomarkers that would be able to actually distinguish different kinds of pain in different populations, because the treatment for one kind of pain may be very different than another, and we do too much lumping right now of pain syndromes. And biomarkers that could actually predict clinical outcomes to a particular therapy. We need objective measurements of pain, what we would call a painometer, instead of the very long in place but not all that useful 1 to 10 scale.
Yeah, so as Francis Collins pointed out, we need biomarkers and we need objective measurement of pain just to facilitate, for example, drug treatment development or evaluation, or patient care or prediction of which treatment will work, and so on. And also to better understand the mechanisms of pain, different pain conditions. But pain, there are many different types of pain, and pain is not that simple, right? In terms of pain duration, it ranged from this acute pain, experimental pain, to really clinical, chronic, long-lasting pain conditions. And also there are many different pain types. I will introduce the studies that I kind of tackled this pain duration thing. So many different types of pain in terms of this pain duration. And we actually started with this modeling experimental acute pain that lasts only around 10 seconds, heat pain. So in this study, we scanned 20 healthy individuals with thermal pain, and each person went through the 12 trials at each of 4 temperatures: warm, low, medium, and high pain. So this was not painful, but warm. And then it had this really classical design of pain study, like there is a cue and thermal stimulation for 10 seconds, and then we got the pain ratings. And so just using this duration, thermal stimulation duration, we got this brain activity brain map for the pain related kind of duration. So, and after we selected some features that are most relevant to pain using meta-analysis, we got these 4 images per person: warm, low, medium, high, like the contrast maps for each condition. And then using these images, we did principal component analysis, and then regression, and we constructed weights, and then we got this map. So this we call neurologic pain signature. This shows some threshold regions that are important predictors for pain, but it actually near these blobs, they have all voxels have the predictive weights, and combination of these predictive weights will we can say the predictive of pain. This neurologic pain signature, NPS, discriminated physical pain from non-painful warmth and pain anticipation in emotional pain and pain recall with really high accuracy, 90% sensitivity and specificity across 4 fMRI studies, and also that was responsive to reduce pain by analgesic drug like remifentanil treatment, and then the signature response goes down, the dose dependent reduction, right? So after the first study, we kept testing the NPS on multiple conditions and studies. And this is an analogy, but we are trying to build this NPS's receptive field that shows the sensitivity and specificity. So to what condition the NPS respond to, for example, activated. We know that the NPS is activated by noxious pressure and electric shock and noxious. This is the training set, noxious hit. But we also observed that the NPS is activated by this visceral pain and other types of bodily pain. And also we tested the specificity. So the NPS has shown that it is not activated by aversive images and social rejection, observed pain and pain anticipation, cognitive reappraisal, and so on, and even the placebo effects. So through this testing, we actually better understand the NPS, the characterized NPS, and But the problem is the NPS explained only 17% variance of trial-by-trial variations in pain, when we averaged across 6 studies. And as you can see, there are so many variants left for further explanation. So using these multiple data sets, we aim to model more variance in the pain ratings. So we use this single trial pain data set. Single trial data sets are quite useful for machine learning, and as I can show you, so in the individual trial by trial stimulus intensity is coupled with the individual's trial by trial brain activity estimates, and also trial by trial pain rating. So input and brain and output, we have all this information, and then we can actually model the brain activity to predict this trial by trial pain ratings. In this study, we use these 4 studies as a training set and these 2 studies as a testing set. And I developed model. Actually, I developed a model per person, 1 model per person, using this single trial model. For doing that, I first regressed out the stimulus intensity effect and also the NPS response, because I kind of wanted to model the variance after removing the NPS, the nociceptive related activity. And then we use the PCA plus combined with regression. We call principal component regression. And then we got the model for subject 1 to 137, and then we averaged them to get this stimulus intensity independent pain signature. So this map is aimed to model the pain ratings explained by not by stimulus intensity, but by the kind of endogenous variations in pain, in individuals. So this is the map, the stimulus intensity independent pain signature, and this is showing the prediction using the originally by MPS, that is gray line, and black dots are the prediction based on both on the MPS and SIPS one, and red one is actual trial by trial pain ratings. So when we combine these two models, we can actually get better prediction, and we can explain a little more variance than MPS alone. So this is the summary averaged explained variance across 6 studies. For the 4 training data sets, we use the cross-validation, and 2 independent data sets, we just use the a priori model. And we see the MPS and SIPS total, we were able to explain like around 26% variance. But there are still, a lot of variance to be explained in the future study. However, when I went through the review process, one reviewer expressed a really deep concern about this work. And this is the quote from the reviewer number 3. It's always a scary name, like reviewer number 2 or 3. And this reviewer mentioned that the paper is very impressive regarding the massive computational technology. Not sure that was massive computational, but anyway, thanks for saying that. But the reviewer really concerned about the technology seems to have convinced the authors that they have discovered a new fundamental brain structural organization rule, where I think they are being misled by statistical tricks. This reviewer really worried about just we didn't actually discover things, but we kind of believe that we found. I don't believe that I found a really fundamental kind of structural rule, organizational rule, but yeah, we found some useful marker, I think. But anyway, one of the major concern of this reviewer was the interpretability. So I really kind of wanted to respond to this reviewer number 3 sincerely, and I kind of has a similar feeling, concern after all, because if the model cannot be really interpreted and we cannot really explain the models with in the perspective of neurobiology, I don't think these markers are going to be really useful. So we started to think that something important is missing in this like vectors for better building better biomarkers, and that was interpretability, we think. So interpretable model and the explaining these models in terms of neurobiology is going to be, I think, really essential. And with Lada Kotova, who is a PhD student in our lab, we wrote a protocol paper recently, and we made a point that why we need interpretable models and the methods that to interpret models. Even in these linear models, there are so many features that are going to be really difficult to understand what each feature or weights really mean. As you know, the deep learning models, nonlinear models, are becoming more and more popular, and it's going to be much more difficult to understand these nonlinear models. But if we do not know why and how a model works, actually we cannot really answer the following questions. When will the model fail, and to which individuals or subgroups does it apply? And how can it advance our understanding of the neurobiological mechanism? And to be interpretable, the model should be A, be readable and understandable to humans and provide useful information about the neural representations and mechanisms of mental constructs and behaviors, and need to show predictions are based on true signal, not noise. So we actually proposed multi-level kind of interpretation framework that consists of these 3 different levels of assessment. One is feature level, so which feature are important for prediction. And model level is how the model behave, so whether these models' behaviors are confound free or is there any bias or weak points or generalizable? And also, importantly, biology level assessment. So based on the previous literature or more invasive studies, we need to kind of put our efforts to get this converging evidence from literature and other types of studies. And if we cannot really understand the biological meaning of the model, we need to test different possibilities that may lead us to novel discovery, and we can maybe develop new theories and new experiments. So this model again, the stimulus intensity independent pain signature 1 model again. So to respond to that reviewer, the really concerned reviewer, we looked at each regions of this model in more detail. So for example, when we looked at the nucleus accumbens, we kind of wanted to convince the reviewer that this is not random and has some meaningful patterns. So when we looked at the nucleus accumbens, we saw some of them have positive weights and then some of them has negative weights. And this was quite consistent with the evidence from human neuroimaging studies. Like previously, Baliki et al. Showed that the shell-like structure, similar to our positive regions, respond to pain onset, and core-like nucleus accumbens region has negative, so respond to pain offset. And so we can think that shell is more like pro-pain region, like respond to pain, and core region is more like anti-pain region, so respond to pain offset. And that's really consistent with our pattern. And also there have been some animal studies that show the nucleus accumbens shells amplifies neuropathic pain. So we can see this kind of nice consistency and converging evidence across multiple studies. But this pattern of like positive versus negative, like gradient like distribution of weights, cannot be observed in this univariate results. So only negative in this univariate analysis. So we argued that this multivariate approach actually can give us some neurobiologically meaningful weights of and patterns, and so that we can actually understand the neurobiology of pain better. So we have these 4 components, and then we updated these components, and recently we using all these components, we tried to model some a little longer pain, so tonic pain. And we used to induce some long-lasting pain in human, like safely, we used hot sauce, so food ingredients. And given that, I mean, when we thought we need to get IRB approval, the tonic pain task itself might not be, I mean, if we just give the kit to a person for a long time, that might not be ideal. And we kind of predicted some challenges in getting IRB approval. And so we kind of started to think that we can use these food ingredients to make the IRB approval a little bit easier. And it's really painful if you try on yourself. So we applied a small amount of hot chili pepper sauce on the tongue. When I was in United States, I used a habanero Tabasco hot sauce, not common Tabasco. That's not really painful, but habanero sauce is really painful. And when I came to back to Korea, I started to use the capsaicin extract. That is food ingredients we can easily buy in a grocery store. Okay, so we know that capsaicin activates the TRPV1 channel, and especially we decided to give that on tongue because the orofacial pain is more like more severe and emotionally draining than body pain, and like more resembles clinical pain. So a very small amount of hot sauce on the tongue induces pain for around 10 minutes, and it stays like really painful for a while, and then gets better and better. And in this study 1, we scanned 5 minutes for this capsaicin condition, and also 5 minutes for control condition. And if the control condition happens later, we gave the screws to soothe the pain sensation induced by capsaicin. So this capsaicin tonic pain test is basically a resting state design with pain rating. And for specificity testing, we use the bitter taste using quinine, that is an ingredient of tonic water, so another food ingredients, and aversive odor, that is also a food type of food that has a really bad smell, like fermented skate. You might not able to imagine what the odor of that is like, and that is like famous food in Korea. In some local area, young people usually don't like it, and actually we before making decision of the what we should use for aversive odor, we kind of did a competition using like cheese and this type of food, and this was the winner, fermented skate. Anyway, we actually made a short video abstract, and I want to kind of share to show how we actually did the study. Okay. And a corresponding author of the paper. Here is the filter paper. Actually, the person who are lying down here, he's Jae-Jung Lee, who is the first author of the paper. Anyway, okay, let's see. With a small amount of capsaicin-rich hot sauce on it. So, in the real experiment, we asked participants to place this filter paper on the tongue. So I will give this to Jae-Jung, and participants lie down in an MRI machine, and please place the capsaicin filter paper. So we ask participants to put the filter paper in the mouth for 30 seconds, and after that we ask participants to remove the filter paper. Can you remove the filter paper? And open the mouth and breathe only through the mouth for 1 minute. This procedure will remove the liquid and settle the capsaicin on a specific area of the tongue. After that, we ask participants to close their mouth, and that will provide some heat to the tongue, and it will induce the painful sensation. Okay, so if you are interested in looking further, you can go to that link, and then you will see 10-minute brief introduction to the paper. And I will introduce that here. Okay, so we had 3 different questions, and first one was can we identify a brain marker of tonic pain based on whole brain functional connectivity? And to that end, we got the functional connectivity features from this fMRI scan. Functional connectivity, we usually define this as a correlation between different regions using the parcellation. We tested many different types of parcellations and different types of methods to get the functional connectivity, because there are many different methods to get this correlation. And the outcome variable was pain ratings, and we used machine learning, many different types of algorithms to generate models. Actually, we got many different models because we had enough data sets, because we had a study 1 data that I collected in the United States, and study 2 data, another data set that I collected in United States. And after I moved back to Korea, Jae-Jung and I collected 33 data set. So we had 3 different data sets, and I used the first data set as a training set, and second data set as a validation set, and third data set as a testing set. So given that we had enough data sets, we kind of generated many different models, and then did an internal competition using the a priori, like predefined criteria. Using this final model, the winner of the competition, we did the independent testing. Okay, so this is showing how we did our model competition within this data set. We had many different options for parcellations and correlation methods and algorithms and outcome variables. And I mean, outcome variables are not like competition, because we can get one model, one winner for intensity, and one model for unpleasantness. But we kind of decided to focus on intensity because I mean, the performance was a little better. But I mean, we had the results on unpleasantness rating in the supplementary material, so you can check. Anyway, and these were the 7 predefined criteria for model selection. So sensitivity and specificity. We only use the study 1 and 2 so that we can kind of hold out the study 3 data. The final winner was the brain atom parcellation and using the dynamic conditional correlation that really take into account the autocorrelation basically. And we tested multiple different numbers of components, and the 21 was the final number of components that worked across multiple data sets. So this is the representation, circus representation of the tonic pain signature. We named it as a TOPS, okay? Fancy name. And I will tell you why we named them. So this is the marker, and I mean, but it's really hard to see what's going on, right? So we did additional analysis and visualization. You will see in a minute. So this model can be applied to independent data sets to test its sensitivity and specificity in an unbiased way, right? So this is the model, I mean, the sensitivity test on study 2. Study 2, we used it for as a validation set. So the performance can be a little bit biased. Just consider that. So, but it shows quite nice prediction, and also the here the red line is signature response and black line is avoidance rating that we got. Why we got the avoidance rating was given that we were using multiple different conditions, like in addition to pain. As I said, we used the quinine and also aversive odor, and we kind of needed a unified rating scheme. So we started to use avoidance rating, how much you want to avoid this stimuli in the future. So like we kind of use that for all these different stimuli. This is the independent data set performance in the independent data set, and the correlation was nice. So this is capsaicin plus control run, so combining these 2 runs. But given that the study 3, actually we scanned 20 minutes per condition, so only for capsaicin. So we actually were able to use capsaicin run only to get the performance. So this is capsaicin run only condition, and correlation was 0.39. And this is the predictive rating across capsaicin and control. There's a little mismatch in the end of the run, but it kind of overall it nicely capture the differences between capsaicin and control, and also the decrease in capsaicin within capsaicin run. And the specificity test, it showed quite nice accuracy. So this is validation set, and these are the independent data set. And the model were able to discriminate the capsaicin from bitter taste and aversive odor with high accuracy. So our tonic pain marker, TOPS, showed good sensitivity and specificity in independent testing. And the second question we addressed was, does the tonic pain signature generalize to clinical pain? So we used this open pain data set, openpain.org. You can get the data from there as well. And Etienne actually contributed this data set, and thanks to Etienne's effort and others' effort in Apokarian lab, and we were able to easily get the data sets and multiple data sets, and then test our marker on these different data sets. So in this data set, there were 2 types of different clinical populations. One is subacute back pain patients, and the other one was chronic pain patients. And there were 2 different task types. One was spontaneous pain rating, where the participants had to pay attention to their spontaneous pain and rate their pain using finger span. Our marker worked, I mean, in this subacute back pain patient who are kind of within 3 or 6 months. I forgot the exact month, but recently they got the back pain, and for them, only when they pay attention to their pain, our model worked. So it explained the individual differences. And in chronic back pain patients, the pattern was a little bit different. It worked in resting state, and a little bit worked in spontaneous pain rating. Like 0.3 is not really super high and not significant given the number of subjects, but it's not 0. So I would say to express the patterns of our marker, they had to pay attention to their pain. But in chronic back pain, even without the attention to their pain, their brain activity patterns or connectivity patterns were similar to our marker to some extent. And also, when they pay attention to their pain, the model worked a little bit. And there were other data sets, actually, another chronic back pain patients data set from Japan and UK. And our marker discriminated patients from control with a reasonable significant classification accuracy. So the TOPS seems to work for clinical pain, at least for low back pain. And one interesting point that I didn't put here was our model worked a little better than the model trained on these clinical pain data sets. So it's an interesting idea that we develop our marker using experimental pain condition, and it generalizes to clinical pain, and actually worked a little better than the model trained using clinical pain data themselves. We kind of had a long discussion about this in our discussion section, but in a nutshell, we think that clinical pain data sets are include many heterogeneous populations, and so our model may capture the sustained nature of pain in a controlled setting, so it has a more homogeneous and more less noisy data, but we need further study to fully understand what's going on there. And last question we addressed in this paper was how similar or distinct are the functional connectivity models among tonic pain, clinical pain, and experimental phasic pain. And this is the another kind of anatomical level representation of our model. We only show the important connections, and as you can see here, really important connections like these and these are all inter-network connections. They were in different membership. This one was frontoparietal network, and this one is somatomotor network and default mode network. All these different network regions within the different large scale networks, they connect to each other for the tonic pain processing. And you can see many, you might think that somatomotor network is really important for pain processing, but what we found is limbic system and frontoparietal network is quite important for explaining this tonic pain signature. And we have the hypothesis that when pain lasts long, these involvement of other brain networks become more important. Because in an early phase, maybe the processing sensory information might be important, but later on, the regulation, top-down regulation, or processing some affective aspects or interoceptive aspects of pain may become more important. So I think we thought this might be similar in subacute back pain. So this is the subacute back pain model, the model that we developed using the subacute back pain data set. And also this is the another data set using the 12.5 second pain. Actually, this data set was collected by Matthew Roy, who is a professor in psychology department in McGill University. Anyway, we use these two data sets and compare these models with the tonic pain signature, and we found that these were similar than the relationship between tonic pain and acute pain, or the relationship between subacute back pain and experimental phasic pain. And these are this plot shows the which is the like closer to which. So this area was the shorter distance between the TOPS and subacute back pain model. So and each dot is the each region, and color shows the networks. And if the more dots are in this area, that means more regions and more voxels are have the shorter distance between the TOPS and subacute back pain models. And as you can see, 7 among these 9 networks were located in this, like showing shorter distance between the TOPS and the subacute back pain model. That means, I mean, overall, our tonic pain signature model has like more, like it resembles this subacute back pain model. So we found that the TOPS is more similar to the subacute back pain model than the experimental phasic pain model. Importantly, why I'm explaining why we named them, and importantly, when we located these models in these different stages, what we found is these really promising models has names. And when we collected some promising candidate markers for, like, for example, Alzheimer's disease or Parkinson's disease, actually they had names, and it was interesting finding. And we after a thought, I realized that naming marker express the intention of the researchers that they want to share and reuse these markers. So we've been sharing many different pattern masks and predictive models through GitHub. And for tonic pain signature, we made the web page to share our model. And I actually already shared our model with Etienne and Matthew. And I heard that Etienne also already tested this model on the UK Biobank data. And I really look forward to see the results. I mean, it's a kind of scary, and I worried about the results, but I think that's a like really healthy fear, right? Of the falsifiability. Our model may work or may not work, and even if our model didn't work on that data set, I want to figure out what is the reason. And I hope that Etienne will share the results with me and figure out what is the underlying reason of whether or not the model works. Recently, I came across this Twitter title was like scientific publication beyond the text and sharing research objects. I really like that, and science, I think science should be more than publication and share the objects and models and codes. So I'm really open to sharing our models and codes and tutorials and so on. Okay, so thanks for listening, and I hope that I can share more ideas and results in the future. And here, Jae-Jung was the first author of the Nature Medicine paper, and Lada was the first author of the Nature Protocol paper. And here, the Thor Rager that who was my PhD advisor, and we worked together through this journey. Thank you so much.